All right, good evening, everybody. So just a reminder, we're covering eukaryotes today in our cell biology section. And after signing in, make sure that you have the strip sort activity close to hand and your mitochondria and chloroplast drawings. If you didn't get a chance to do these, it's just going to be what it is. But if you have them available, it will help you in the class. So kind of a laundry list of what we're going to cover. Cell wall composition in eukaryotic cells. This is not peptidoglycan. Plasma membrane function. We're going to just look at a couple of differences with prokaryotes, though there are many similarities. Flagella and cilia. What is their structural differences and molecular differences? Ribosomes that have that different density. And then we're going to talk about the endomembrane system, how it works. And then we'll finish up with mitochondria and chloroplast structure. That's super helpful for us to review before we hit metabolism in Unit 2. And then the endosymbiotic theory. So this is a picture that's probably pretty familiar, right? You've seen some version of this in any cell biology text. So just tell me what cell type is on the right versus on the left. Put into the chat. What are the two cell types shown here? Put that into the chat. What are the two cell types shown here? One's on the left, one's on the right. Even if you know just one of them. All right, Megan, no problem. And don't worry if I forget and call on you. I won't take it personal if you're not answering. Hopefully you don't take it personal that I forgot. You just told me that might happen. Okay. Yeah. So on one side, we have some kind of animal-like, potentially protozoan eukaryotic cell. And on the other, we have a plant, or could be an algae, right? But it's definitely a eukaryotic cell. We've got all sorts of organelles, the nucleus. What I'm going to do really fast is just kind of a quick review. And just like we did with a bacteria, only with lots more line items, I'm going to show you a picture and I'm going to call your name and you're going to say what you think it is. You can always say pass. That is not a problem. But just to sort of warm up today, we're just warming up. Okay. So starting with A1, the thing that is A1 is pointing to this part of the cell. So for that, we're going to go to Amy. What do you think A1 is? Or would you like to pass? So give us your guess, or you can pass. And I'll watch the chat in case anyone has microphone issues.
I will pass.
Okay, no problem. Ayumi, what about A1? What do you think? Are you able to unmute Ayumi?
Sorry. Nuclear envelope?
Yes, that's a good guess. I agree. That's what it looks like. All right, for A2, just the way these lines are looking, it's a little hard to see. So A2 is pointing to these holes, okay? These holes. Dorothy, do you have any idea what these holes might be? Do you have a guess or do you want to pass? Either is fine.
The nuclear pore?
Correct. Okay, let's move on to Gabby for A3, all that squiggly stuff.
That the DNA?
Yes. And then A4, let's go to Haley Gonzalez. Haley, what do you think A4 is?
That looks like it's pointing at the nucleus.
Nucleolus, yes, indeed.
Nucleolus.
Yep. I knew that's where you're going. Just helping out a little bit. And all together, all of this, all A1 through A4, guys, into the chat, what's A pointing to in general? Put into, yeah, exactly. This is the nucleus, obviously the defining feature of the eukaryotic cell. All right, for B is pointing to several things that don't seem to all be the exact same size, but they're all filaments. So B, Janelle, what do you think B is pointing to?
I think cytosol.
That's close. That is close. Does anyone want to raise your hand and help her out here? It's a word that's very similar. Tiffany, go ahead.
Is it like the cytoskeleton?
That's right. Good job. Okay, so we can see some skinny things and some tube things, probably microtubules, maybe actin or intermediate filaments. Excellent. Okay, Jenica, what do you think C is pointing to? It's this whole area here encompassed by this bracket.
I would say rough ER.
So part of it is rough ER, but it's going the whole way across. What do you think? And if you're not sure, that's okay.
Yeah, I'm not sure.
No problem. Okay, anyone else want to take a guess? You want to raise your hand and say what you think it is? Rough ER certainly in there. Dal, what do you think?
Is this the microchondrion?
It's not, but that's an excellent guess. Anyone else want to take a guess? Amy.
I'm gonna guess the plasma membrane.
That's actually not correct, although plasma
No.
membrane is in there too. So good guessing, guys. Anyone else? Lori, what do you think? This is fun.
Plasm,
Say again.
cytoplasm.
Yeah, so one way to interpret that, which I would agree with, is cytoplasm, sort of encompassing the whole area. Yes. There is another way to look at this. You could, and this is not how it's in the key, this is not what it's shown, but if someone were to think, oh, well, to me, it looks like it's the endomembrane system because so many things in the endomembrane system are a part of this, I would accept that too. All right. Though it's not really important to get like to study for this for a test. We're not going to show you this picture and have you do this. We're just warming up, trying to remember all the stuff we know about the cell. All right, what is D? D pointing to John. What do you think? Yes, he's in the chat. It's the Golgi. That would be a good answer. E, there's this pocket here. You can see that it says, assume that it is acidic. Caddy, do you have any ideas what that might be? You can unmute and or write into the chat if you have a guess, or you can pass. Either is fine. Okay. A couple people are guessing. Caddy says pass. Okay, great, Caddy. Thank you for that. Kim, I'm going to ask you what you think. And if you want to pass, that's also fine. But what do you think this is?
Is it the vacuole?
It is a form of something that could be considered that, but it's a little bit more specific because it's acidic, and in this cell type would be something else. That's a good guess, though. Excellent guess. Let's see. I'm going to pick one more person. Megan, I'm going to skip over you for right now. Just give us a heads up when you're able to join in. Miliana, do you have a guess as to what it might be? She's not sure. That's no problem. Anyone want to raise your hand and give another guess? Tao, go ahead.
Is it the vesicle?
It is a kind of vesicle. It is more specific, though. Most vesicles are not acidic on the inside, so we're getting closer. Good job. Ayumi, what do you say?
I guess the lysosome.
Yes, it's the lysosome with the acidic interior. Good job, you guys. All right, now look at what's happening here. This is a process where stuff is, there's a vesicle fusing to the outside that's dumping its contents out outside the cell. What process is depicted here? For this, I'm going to ask Alex.
Exocytosis.
Yes, that would be a good guess. G, what's depicted by G, Leslie? Are you able to hear us and unmute Leslie or write into the chat? Okay, I'm going to ask Surmeet, what do you think is G?
I believe it is ribosomes.
It is a ribosome. All right, H. For H, I'm going to ask Sarah, what's H?
The plasma membrane.
Yes, plasma membrane. And I, for Nikki, let's go for I.
I'm just guessing, is it the mitochondria?
It is the mitochondrion.
Okay.
Excellent guess. All right, Jay. Now we're just going to start to see if anyone has a hand they want to do. So raise your hand if you want to guess on Jay. Jenica, go ahead. You're still muted.
I'm sorry. Is it smooth ER?
Smooth ER is a good guess. All right, K. K. Anyone want to guess on K? I feel like this is sort of a weird bingo. Anyone want to guess on K? Lori, go ahead.
Is it a vesicle?
It is a vesicle. And L, of course, what do you guys think? Let's have everyone write this into the chat. What's L? What's shown by L? L is pointing to what? Yeah, everyone knows this, rough ER. And M, you can also put M into the chat as well. Flagella. Good job. Okay, so just to remind you, this is not cell biology class. I'm assuming you all know this stuff. So this is just a warm up. Okay, so we're not going to have like this picture on a test or anything as straightforward as that. What cell type are we in now? That was clearly either an animal or some kind of protozoan. What is this? And there's actually two options that would be good. Yeah, it could be a plant cell, probably is a plant cell. It's also possible it's an algae. Some algae are multi-celled, though they're not macroscopic. They have a few cells, so it's possible. We see a few structures we did not see in the previous. So number one, guys, what's that? Put that into the chat. Chloroplast, right? Photosynthesis is happening there. And then what's this giant bag of water here? Number two, what's that? Central vacuole, central vacuole. And last but not least, we also see this kind of thick border, which of course is what? Cell wall, correct? And what's it made of? That's right. Good job, is cellulose. Cellulose. Plants and algae have a cellulose-based cell wall. Okay, so again, this information, if you want to review it, if you're feeling like, oh, I was super rusty, is on Canvas. There is this diagram. There's an answer key. You can look up anything you want, but I'm not going to be showing you a picture like this on a test and asking you just to identify it visually. I'm assuming that you know these structures and what they're for. So if you're not sure, just have a quick review. What I want to do, like I do, is just, again, demonstrate what electron microscopy brought to the table in the middle of the last century when it revealed the inner workings, the detailed inner workings of cells, including eukaryotic cells. So, you know, some of this stuff we'd seen in some way, shape, or form before, but under an electron microscope, there's so much more detail. Okay, and we're going to kind of skip over the guessing part of here, but or a comprehensive one. But I'm just going to give you a second to eyeball it. And if there's something in here that you feel like either super confident you know what it is and want to say, I think it's this, or you're just really curious to know what it is, then just raise your hand. So take a look at it. You know, if there's anything you want to comment on or say, I think this is this, we'll take a second to do that. Appreciate all the incredible detail that is afforded by electron microscope images. All right, anybody want to raise your hand and say, I think something, something, or I'd like to know what this thing is? Okay. The weather's too nice for this activity. That's fine. Me personally, I think the coolest thing is these little cucumber things, which of course are chloroplasts, and these are the mitochondrion. And I'm pointing those out in particular because we will spend a bit of time talking about those, and we can kind of see some interesting architecture inside of them. But here is an answer key for those of you who might have been curious and just feeling a little shy to see if you were right about your guesses or explore your curiosities. Again, I'm not showing this on a test for you to identify and label. We're going to do more things than that. So one of the things we have to tackle now, we're going to kind of go through the eukaryotic cell, and we're going to do a lot of compare contrast with the prokaryotic cell that we covered on Monday. So starting with the cell wall, the function is pretty much the same, right? This function of the cell wall is protection. Which eukaryotes, which eukaryotic microbes in particular, but which eukaryotes in general have a cell wall? Yeah, plants, algae, and fungi. And of those, algae and fungi are the only ones that are microscopic. Yeah. And so notably animals like ourselves and protozoans, which are the single-celled animal-like members of kingdom Protista, these do not have a cell wall. Now the cell wall composition is distinctive between groups of microbes. So plant-like things like algae have cellulose as their cell wall, and the fungi, the microscopic ones and the macroscopic ones, but the microscopic ones is the ones we care about, like yeasts and bread molds and things like that. Their cell wall is made of chitin. And both of these are very similar molecules. They're polymers of glucose. They're just stuck together in different ways. And the importance of knowing what kind of organism you're dealing with is that the cell wall may or may not be a good target for drug treatment, right? Penicillin was called a miracle drug. It doesn't harm people because it targets the cell wall of bacteria. It specifically targets the peptidoglycan crosslinks. And so we're not going to be able to use penicillin to treat things that don't have peptidoglycan. So can you use penicillin to treat, let's say, a toe fungal infection? Right, and why not? The answer is no. And probably, if you think about it, you've never had anyone prescribed penicillin for a fungal infection, but why not? Yeah, the cell wall is made of chitin. It's not made of cross-linked peptidoglycan. And in fact, treating fungal infections, you might know this, it's trickier than treating bacterial infections. The types of drugs we might choose are likely to have some toxicity. We have to be more careful because there's more similarities between our cells and fungal cells than there are between our cells and bacterial cells. There's not as many levers that we can pull. Okay, now another difference, so sort of similarity and difference we have to talk about is the flagella and cilia. Now bacteria, they had flagella, right? And the function of the flagella in bacteria was motility. What is the function of flagella in eukaryotic microbes? Yeah, also motility. So the function of both flagella and cilia in microbes, in eukaryotic microbes, is motility. Now, if you get into multi-celled organisms, there can be more complicated answers to that, but we're focusing on microbes. Now, what is a eukaryotic flagella and cilia are the same structure, actually. What are they made of? Put that into the chat as much as you can recall. And this is the eukaryotic flagella and cilia. So make sure that your answer is focused on the eukaryotes, not the prokaryotes. Yeah. So many of you mentioned that the flagella in eukaryotic cells and the cilia as well are made of microtubules, these long filaments that are kind of hollow, like straws. You can think of them like straws, but it's not just one of them. It's actually what we call a 9+2 array. So if we chop one of these and we look down the barrel, the cross section shows us this interesting organization where we can see 9 pairs of microtubules that are all running the length of this thing and 2 in the middle. So 9 pairs plus 2 in the middle. What you can't see is that there's motor proteins connecting these that are moving these microtubules with respect to each other. And that's what causes the motility of this flagellum. And the motor proteins are all along the shaft of the flagellum, or the cylinder. And this is very different from bacteria, where bacteria have a single motor at the attachment point that spins, right? So the motion, if you were to view it and describe it in words, is not rotational. What is it for eukaryotes? Whip-like or wave-like, right? And if you have trouble remembering this, just imagine a human sperm cell. That's an iconic video that almost everybody has seen. And you're just seeing it like its tail is kind of whipping or waving or, you know, thrashing, or however you want to describe it. But the motion seems to occur all along the entire length of this flagellum. It's not coming from one spot at the attachment point, which is, of course, different from bacteria. Now, prokaryotic flagella are for motility, but they are made of flagellin protein and they rotate. And it's helpful, I think, to kind of have a sense of scale here, right? Remember what the size of the thickness of the prokaryotic flagella, right? A bacterial flagella is how thick? What's the width of it? 15 nanometers, right? One microtubule is 25 nanometers. So the bacterial flagella would fit into one of these 9+2 array of microtubules. And then you've got these two and this. So this is obviously a much, much bigger structure, which explains why you can see it under the light microscope under the right conditions. Furthermore, cilia, which appear on this picture, for example, like little hairs, they're shorter, there's lots more of them. At a first glance, if you were to look at this picture, what does it look like to you, or that we covered last time? It resembles what? These little hair-like projections. In bacteria, what is a structure that kind of resembles this? Yeah, pili or fimbriae. But remember, the thickness of this cilium is like this compared to the pili, which is, again, is going to fit into just one microtubule. Not to mention that we're sort of seeing like this is the size compared to the whole cell. And it only looks similar because of scale. Because remember that the prokaryotic cell is much, much smaller than this, right? And then so we have to understand that the fimbriae and pili are also much, much smaller. And unlike in bacteria, flagella and cilia are made of the same thing. They look like this. Whereas in bacteria, flagella are made of flagellin and pili and fimbriae are made of pilin. All right, is there a glycocalyx? In bacteria, it was either capsule or slime layer, but, and that was made of sugar. Do eukaryotic cells have a glycocalyx? Yes or no? Yeah. Yes. Often in an organism like ourselves, that is part of the sort of layer that is, you know, the mucous membrane, and it's often the first line of contact with a pathogen. All right, eukaryotic cell membrane. Now, what is a common function that the eukaryotic cell membrane shares with the prokaryotic cell membrane? We talked about the function of the cell membrane, and in prokaryotes, there were a couple of things. What function do these two membranes share, or what do they have in common? Write that into the chat. Yeah. Yes. Yeah. The membranes behave like membranes. They have selective permeability. They allow the exchange of nutrients and waste. Therefore, they get information, communication, right? So that's in common. Now, structurally, there's also commonalities. What is the structure of the eukaryotic cell membrane that's similar to the prokaryotic cell membrane? Yeah. Mm-hmm. That's right. So it is a phospholipid bilayer and has membrane proteins in it. Now there are some differences. Raise your hand if you know of any differences between these two membranes, either things that the prokaryote can do that the eukaryote can't, or the eukaryote can do the prokaryote can't, or structural differences. But let's start with functional. Gabby, what would you say is different?
I have that in eukaryotes, they have or do endocytosis.
That's right. The bacterial membrane does not endocytose. So what that means is the exchange is limited to single molecules that can be handled by a transporter. When stuff comes in to the bacterial cell that is something that doesn't cross the membrane on its own, that needs help, then what's helping it is a protein-based structure. Okay, so it's going to only be able to bring across relatively small things, ions, single sugar, like a monosaccharide, one amino acid. So that kind of makes, there's a limit on how fast you can do that and how much you can bring in at once. In the eukaryotic cell membrane, there is the ability to do endocytosis, where you can take a much bigger sort of chomp out of the world, and you can bring in not just one amino acid, but multiple proteins. So like, you know, maybe 100, maybe even 1,000 times more stuff all at one time. And that's kind of convenient because don't forget that the eukaryotic cell is much larger. And so we need to have some way of boosting our exchange power. Now, the other thing that's different is, remember we talked about how prokaryotes have put their respiration and photosynthesis machinery, some of it's in the cell membrane, right? Because they don't have organelles. But that's been delegated in eukaryotes to these mitochondria and chloroplast organelles. So the ATP synthase and all the associated bits and pieces like the electron transport chain and any photosystems, they're not in the membrane anymore. Now, structurally, there also are differences, right, having to do with the presence or absence of steroids. For example, you may know this, animal cells have cholesterol, and fungal cells have ergosterol, and plants have phytosterols. These are not found in bacterial membranes. Okay, question for you. We're going to talk about the cytoskeleton. Do prokaryotes have a cytoskeleton? We know that eukaryotes do. Do prokaryotes also have one? Right, they do not. If you have trouble remembering this, just again, think about scale. Think about how enormous a microtubule is compared to like, you know, even a structure as simple in the bacteria as a flagella. So there are no cytoskeletal filaments in prokaryotes. Now, in eukaryotes, the cytoskeleton is composed of 3 major types of filaments, and there's lots of different things they do. What is kind of, just list one thing in the chat that you know that cytoskeleton does, because it's lots of different things. There's 3 different filaments, so some do some things, some do others, but we get, yes. So cytoskeleton can support this shape of the cell. Microtubules provide like sort of gross shape, like overall shape, and microfilaments or actin can sometimes provide small detailed shape. They're also responsible for movement in lots of ways, actually. So of course, microtubules are part of flagella and cilia that are involved in movement, but actin's involved in crawling motion. Interior organization. Microtubules are basically like a scaffold that the inside of the eukaryotic cell hangs on, which is handy because there's a lot of stuff in there. We actually do need to organize it. Okay, anything else that cytoskeletons do that you know we could add to our list? Okay, let's have a look. So cell shape and movement, that's right, Jenica, cell division. So cell shape and movement, organelle organization and movement of organelles along the highways of the microtubules that run through the cell. And then cell division, also facilitated by the mitotic spindle, which is made of microtubules. Overall, how would you describe the type of chemical that makes up the cytoskeleton? Yes, that's right. It is made of protein, lots of different types of protein. Microfilaments are made of actin, microtubules are made of tubulin, and then intermediate filaments are made of a variety of things depending on the specific cell type. And we can see a little chart of that here, kind of looking at the different sizes and the different organizations, and then talking about the different things that they do. So here is a kind of microscope view of that. You can not easily see the cytoskeleton under a light microscope. You start to see it with an electron microscope. But what you can do is use a fluorescence microscope to light it up. Okay, so this picture is derived, it's a little cartoon, but it's based on taking a cell and literally causing the actin to light up and turn red so we can see where it is. Okay, so this actin has which of the following, where does it fit in this little chart? Which of these functions best describes what actin is doing? The middle one, the left, or the right? If you're not sure, you can always put a question mark, but do engage with the question in the chat to strengthen your understanding. It's fine to guess. You don't even have to be right. I'll give you another second to speculate. And question marks are all welcome. Yeah. Wonderful. So we have lots of disagreements. Some people are saying they have no idea. Some people think it's the left. Some people think it's the middle. I would say so far, nobody has guessed the right-hand side, which is fine because it's not the answer. But if we look at where it's distributed according to this cartoon, we can see that it's at sort of perpendicular angles. The filaments are crisscrossed at the edges of the cell. And that's because in some cells, they facilitate crawling motion. So movement of the whole cell and local shape, little fine detailed structural things, is what actin does. It's the one on the left here. And what is its size? Write that into the chat if you remember the size. Oopsies, I just revealed it. 7 to 8 nanometers. That was a teacher fail. All right, now here we have two options left. We've got this type. So this type grows from a central organizing center, and it kind of grows out from the center of the cell and reaches out to the periphery in a meshwork, kind of like a series of roads. Which type is shown in this picture? Again, if you're not sure, you can put a question mark. You can take a solid guess. You get 50% chance of being right, even if you have no clue at all. Great. So this one is the microtubules. And they provide internal transportation and organization, and of course, also cell division. And they're the biggest ones. They're 25 nanometers wide and they're hollow inside. And that leaves just one, right? That looks like a rope. And it's found kind of in this, it's not at the edges, it's sort of perinuclear, but also in the cytoplasm. And so what are we looking at here? What's that third kind called? Put that in the chat. Question mark if you don't know. I'll wait. Yes. The third type is intermediate filaments. They're up to 10 nanometers. Okay. Now we have to talk about ribosomes because prokaryotes have them, eukaryotes have them. There's similarities, they're different. So a similarity, protein synthesis, eukaryotic ribosomes do that too. A difference is the structure. So prokaryotic ribosomes have two subunits, the 30S and the 50S, which together makes a 70S. What do eukaryotic ribosomes? What's their structural organization? That's right, Ayumi. Yes, Gabby. There's just straight up memorization. Yeah. So eukaryotic ribosomes are 80S, and they're and that includes a 40S and a 60S. So the small and the large are slightly more dense, and overall the ribosome is more dense, though again, it's not a linear unit. So 3, 5, 7, 4, 6, 8 is how you can easily remember it. There's another difference where these ribosomes are found. In prokaryotes, there's only one place for ribosomes to be, in the cytoplasm. But in eukaryotes, they can be in the cytoplasm or they can be attached to the rough ER. Question. Are there any 7DS ribosomes in eukaryotic cells? Think carefully and answer. Great. The answer is yes. Raise your hand if you want to tell us where they are. Sarah, go ahead.
In the chloroplast and the mitochondria.
Yeah. Chloroplasts and mitochondria are interesting and unusual because they have their own ribosomes. And those ribosomes are prokaryotic. They're 7DS ribosomes. So if you've got a mitochondrion or a chloroplast, which is common, though not found in every eukaryotic cell, then you've got 7DS ribosomes inside of you. Okay, we are not going to take a break right now. We will take a break eventually. So I just want you to write into the chat name any eukaryotic organelle you can remember. I'm going to read them out. Nucleus, peroxisome, Golgi, ER, mitochondria, vesicle. Okay, so someone said ribosome. I thank you for saying that. Ribosomes are not an organelle because they don't have a membrane around them. So that's annoying, right? Because they sound a lot like the other isomes. You'd think they'd be organelles, but they're not. Lysosome, yes. Centrosome, similarly, a centrosome is not bounded by membrane, not considered an organelle, though it is a structural component that's important. Rough ER, distinguishing it from smooth ER, vacuole. Yeah, good. So a lot of great answers. You remember some of these guys. Now, here's a list, not comprehensive, of some of the famous organelles and kind of just remembering us, what do they do? The nucleus contains the chromosomes. The ER is a transport network. This is really vague. We're going to talk more about this. Golgi makes membranes and secretes proteins. The vesicles transport things between the Golgi and the ER primarily, and then the plasma membrane. Lysosome is digestive mitochondrion, is where respiration happens. Chloroplast is where photosynthesis happens, and there's lots and lots of other ones. But today, we're going to mostly focus on the following two categories, I would say. In purple, the endomembrane system, and in red and green, the energy generating mitochondria and chloroplast. So mitochondria make energy, and ATP is the final product of cellular respiration by breaking down things like glucose from pyruvate from glucose. Chloroplasts make ATP, but that ATP is then used to further synthesize glucose. So we're going to begin by talking about the endomembrane system and kind of giving you a sense of the function of each of the members of this system and how they're connected to each other. So of course, the nucleus is where the genes are transcribed to mRNA. And those mRNAs are sometimes intercepted as they're leaving through the nuclear pore by a ribosome, so that the rough ER ribosome can make protein that ends up being usually secreted or put into the membrane. The smooth ER makes lipids, which is important too, partly because as we're moving stuff around, we continually are pinching off membrane and adding membrane. So we need to make lipids to grow the cell membrane, for example, also makes things like sterols. Vesicles transport protein and lipid to the Golgi from the ER, where they become modified. The biggest modification is for anything that's going to leave the cell. It gets decorated with sugars. And then vesicles further bring those items to the membrane where they're released. It is important to know that the lysosome is considered part of the endomembrane system, as well as being a product of the endomembrane system. It's made from the Golgi, and we'll look at those details. But here is basically what the endomembrane system does. All those organelles all collaborate together to make proteins that are going to leave the cell, proteins that are going to stay in the membrane, lysosomes that will stay in the cytoplasm and digest stuff, and any resident ER and Golgi enzymes. So I need to ask you a question first, okay? And in asking this question, I'm hopefully reminding you, or maybe illuminating, that in a eukaryotic cell, there are, there's ribosomes in the cytoplasm, there's ribosomes on the rough ER. Does the endomembrane system, which we know makes proteins, does it make proteins that are going to stay in the cytoplasm of the cell, such as glycolytic enzymes? Yes or no? Now for this one, you cannot pass. Just make a vote, even if it's a total guess, and see where we stand to begin with. Does the endomembrane system make all the protein basically in the cell, including the cytoplasmic protein? Yes or no? Great. Now, before I answer this, I just want to tell you that it was a while into my degree, my bachelor's degree, before I knew the answer to this question. Because I was taking a biology degree and learned lots of different things. But it wasn't until I actually took cell biology and then subsequently more detailed cellular and molecular biology that I really learned the answer to this. I hadn't really thought too much about it. You know, we learned that what the ER does is it makes proteins, and then it passes those proteins off to the Golgi, which sorts them and then sends them where they're supposed to go. So it's natural to think that is kind of where all the proteins get made. That is not the case. So proteins that are going to stay in the cytoplasm, like enzymes that are doing cytoplasm-y things, like the glycolysis enzymes, those are just made by ribosomes that are in the cytoplasm. They're not made in the rough ER. And that's because the rough ER is actually needed specifically to make proteins that are going to need something special. They're going to leave the cell, maybe they're going outside, or they're going to go to the membrane receptors, or maybe they're going inside the ER or inside the Golgi or inside a lysosome. But those other regular guys that are just hanging out in the cytoplasm, things like actin protein and tubulin protein, but also enzymes, those are made by cytoplasmic ribosomes and they never go through the ER. Now, most of you said yes when I asked you this question. So that indicates that, like me, you were kind of fuzzy on that. Okay, so we're going to talk about it a little bit. So I'm going to give you kind of like the visual walkthrough of this before we take our break. So in the last few minutes before the break, we're going to do a visual walkthrough, and then I'm going to leave you with kind of instructions on what we'll do with our breakout when we get back. So protein synthesis. When we talk about cytoplasmic proteins, what's involved? And by cytoplasmic proteins, I mean, here's the cell, proteins that are going to hang out for their whole life and do their job here inside the cytoplasm. Here's the nucleus. There's DNA. The DNA has genes in it. Let's suppose there's a gene over here for glycolysis enzyme. Then we transcribe it into mRNA. It leaves through the nuclear pore. It's found by a ribosome, which translates the mRNA into protein right there in the cytoplasm, and bam, you're done. Okay. So this is quite straightforward compared to what happens to all the other things we're going to talk about. Let's suppose we want to export a protein. We're a B cell. We make antibodies. We want to export them for the whole body to enjoy. Or we're a membrane protein. We need to put a protein somewhere on this membrane here of this cell. That has to go through the endomembrane system. And what begins is first in the nucleus, there's the DNA instructions for that protein, whatever it is. It's transcribed into mRNA. That mRNA leaves through a nuclear pore. It's grabbed by a ribosome in the cytoplasm. But as it is translating, the actual polypeptide that's emerging from the ribosome has some kind of information that tells the ribosome, I need to go to the ER. So off it goes to the ER. It finishes making the protein in the either lumen or the membrane of the ER. And then now the protein is in the ER instead of in the cytoplasm. What happens next? Well, the ER is this complicated network of membranes. And wherever that protein is, it gets pinched off into a vesicle, right? And that vesicle travels to the Golgi, it fuses with the Golgi, and it dumps whatever it's brought with it into the interior of the Golgi. And then we have the Golgi does some kind of modification. It recognizes that's a protein that needs to go to the surface, modifies it with sugar, process called glycosylation, and then packages it up into yet another vesicle to be sent to the plasma membrane where it's exocytosed. Now, if the protein is a antibody, let's say, or something else that needs to just leave the cell altogether, insulin maybe, if this was an islet cell of the pancreas, then what would happen is that that protein would be entirely in the lumen of the ER. It would then be fused with the Golgi and be entirely in the lumen. That's the interior, fluid-filled interior of the Golgi. It would then be in the interior of this vesicle and be dumped outside and would just leave. But if it was a membrane protein, it would be made into the membrane of the ER. It would then end up in the membrane of the vesicle, which would then fuse with the Golgi and put it into the membrane of the Golgi, which would then decorate it, and it would end up in the membrane of this outgoing vesicle, which ended up fusing with this, and anything here on the surface in the membrane of this vesicle is going to end up fusing with the surface of the cell. Now, I know that was a lot, but I'm just going to recap it in words. If there's a protein and it's the point of that protein in the genome is for it to be exported, like an antibody or like insulin, something like that, a protein hormone, then the gene for it is transcribed to mRNA in the nucleus. The mRNA goes through the nuclear pore, ends up in the cytoplasm. It is translated into protein inside the ER. It's then packaged up into a vesicle and sent to the Golgi. In the Golgi, it is decorated with sugar. This is the process of glycosylation. And then packaged up into yet another vesicle that is sent to the cell membrane, where exocytosis releases the contents of the vesicle. So we have 1 minute before the break. And so I'm going to ask you a question, and then I'm going to start here just to kind of review to see if we remember. I'm going to ask you which system made this protein. Was it an endomembrane ribosome or a cytoplasmic ribosome? You're a B cell. You make protein antibodies. To make these antibodies, do these antibodies get made in the endomembrane or in the cytoplasm? Put your answer into the chat. If you have no idea, you put a question mark, but you must engage with this activity. And then we'll take our break as soon as we have gone through my list. Yeah, many of you recognize that it is through the endomembrane system because it's a secreted, antibodies are secreted. We have to go through the endomembrane system. What if you're a T cell? You have a membrane protein on the surface like CD4 on the surface of a T helper cell. So you've got to get proteins to your membrane. Are you going to send them to the endomembrane system or you're going to make them in the cytoplasm? Excellent. So most of you said cytoplasm, and that is incorrect. So membrane proteins go through the same process. There is a subtle difference in how exactly they're inserted into the ER, which we'll talk about. But if you want a membrane protein, if you want something in your plasma membrane, got to go through the endomembrane system. Let's suppose there's an enzyme that needs to go to the Golgi because it's going to do its job there. It's one of the enzymes that glycosylates things in the process of the endomembrane system making stuff to go outside. So we need this enzyme inside the Golgi. Does that mean we make it in the endomembrane system or in the cytoplasm? Okay, so a little bit split here. So this one is definitely endomembrane. So you want to end up in the Golgi. So you're going to start by going through the ER first and then being sent to the Golgi. What about a glycolysis enzyme that's in the cytoplasm, right? One of these enzymes that breaks down glucose in the cytoplasm, that's where it's going to live its life. Is it made by the endomembrane system or by cytoplasmic ribosomes? Right. Finally, one that's made by the cytoplasmic ribosomes, yay. And I'll leave you with one final thought that also I didn't know when I was in, I don't know exactly where I was, but I was in the middle of my degree and I was like, oh, I have this backwards, or I had made this wrong assumption all this time. I thought that there were two populations of ribosomes, the ones that live in the cytoplasm and the ones that live on the rough ER. But actually all ribosomes are the same. They start out in the cytoplasm. It's the protein that they are making that determines whether they just hang out in the cytoplasm and finish there or end up going to the ER to deliver the protein to the ER. Okay, so on the break, you can make sure that you go get your strip sort. This is the basic strip sort activity. There are 6 possible steps, not all of which are used in every situation, but these are 6 possible things that can happen in the endomembrane system as stuff goes from ER to Golgi to plasma membrane, or ER to Golgi to lysosome or whatever. Okay, so what you need to do for each of the different scenarios that I'm going to lay out, you need to figure out which of these steps is required in which order. And the first one that you're going to do when we come back, we'll do this together as a class, is the antibody one. We're going to talk about if you were a B cell and you needed to make antibodies, what would you do in what order? So that's where we're going to begin after the break. So I will see you in just over 10 minutes, at 5 minutes after the hour, and we'll do this together. Okay. See you then. Okay. All right, so we're back. I would like you to take a minute to look at these different things that happen in the endomembrane system and take your best stab at which things have to happen in which order. If what your goal is, let's suppose you're a B cell and your goal is to send an antibody, which is a protein, outside of the cell. Put your numbers into the chat once you've decided what they should be. If you have no clue at all how to do this, you can always put a question mark, but do engage in some way, shape, or form so I know not to wait for you before moving forward. And do your best. It doesn't have to be right. Just look at it and see what you think. Okay. So fill in the last, put in your last guesses. If you're a B cell, your goal is to secrete antibodies, protein antibodies outside the cell. Which of these 6 steps, not all of them, but which of these 6 steps in what order is required to accomplish that task? So I'm going to reveal the answer and then talk you through it. Okay. So the first step is the genetic information for that antibody gene has to be made into mRNA. Of course, that happens in the nucleus. And then the translation into protein has to be completed in the ER lumen. And lumen is a word that means the inside of, or the inside fluid-filled compartment of some closed thing. So inside of the ER, in the sort of the fluidy, floaty part, we've got to put that protein inside of it. Now there's a protein inside the ER lumen, and you want it outside the cell. So what do you do next? You package it up into a vesicle that goes from the ER to the Golgi. And it's in the inside of this vesicle, right? So it gets packaged up, surrounded by membrane. That vesicle goes off to the Golgi. It then fuses to the Golgi, and it's dumped into the Golgi lumen, right? The inside of the Golgi. Since it's going outside the cell, it has to be decorated with sugar. That happens with the glycosylation enzymes in the Golgi. That then tells the Golgi, oh, this is going to the surface of the cell. So it's packaged into another vesicle that's destined to the plasma membrane. And once that vesicle reaches the plasma membrane, the membrane of the vesicle fuses with the membrane of the plasma membrane, and the contents of the vesicle are dumped outside. If you don't believe me, let's go back and look at it, right? So we're making our mRNA. The mRNA is picked up by a ribosome, and eventually the ribosome translates that protein into the interior space of the ER. That's packaged up into a vesicle. It's sent to the Golgi. It ends up in the interior of the Golgi. It's glycosylated there. It's then packaged up into another vesicle. And then that vesicle, when it fuses to the membrane, its contents get dumped outside. Okay, so with that understanding of how this works and what we're trying to accomplish here, we're going to do another one. And for this one, I'm going to send you to breakouts to talk to other people. I'm going to give you a minute to think about it before you go. All right, thanks, Megan. I'll give you a second to think about it before you go away, so you have something to talk to your people about. But what your goal is now you're a T cell. Okay, you're a T helper. One of the things in the membrane of T helper cells is proteins that belong to the CD4 complex. So imagine you're trying to make proteins for the CD4 complex. They're going to reside in the T helper cell membrane. You need the endomembrane system to get those proteins to the surface and embed them in the membrane. So think about which of these steps you would use. Hopefully you have your strip sort in front of you. So when I send you breakouts, you can refer to that. But if not, you can take a screenshot because it's right here, so that you know what you're trying to accomplish. I'm going to send you out in groups of 3, and you're going to talk to each other about what you think. And when you come back, if you're done, whenever you get done and you think you've sussed it out, just come back on your own to the main room and put your answer into the chat. And then I eventually will close the rooms. Okay. So I'm creating the rooms now. Get your screenshot if you need it. Again, what's your job? Which of these steps in what order are required to get proteins into the membrane? All right, welcome back, everyone. Go ahead and take a second to put your answer into the chat if you have not done so already. And if you didn't get all the way through it, you can just put in as far as you got. Even if you just had one number or a guess, you can do that. Okay, let's have a look at the answer. So it actually turns out to be extremely similar to the first one. Okay, and I'm gonna go back to the cartoon in a second to hopefully illustrate that. But really, there's only one difference. The only difference is when you very first make the protein, instead of putting it in the like fluid-filled interior of the ER, you insert it into the ER membrane. As a consequence, when that protein is packaged up into a vesicle, it is not in the inside of the vesicle, it's in the membrane of the vesicle. And when that vesicle fuses with the Golgi, it's not in the interior of the Golgi, it becomes fused to the membrane of the Golgi. It does get glycosylated and then sorted and packaged and sent to the plasma membrane. And when it joins up with the plasma membrane, it's in the membrane. So I'm just gonna go back. Let's see. And go back to the cartoony thing. And illustrate that. So we see that if we want to make a protein to go in the membrane, we want it to end up here, okay? So first we make the transcript, it leaves to the nuclear pore, it's grabbed by a ribosome. When that ribosome is translating the protein, it sticks it into the membrane of the ER. So when you make a vesicle out of that membrane, it's in the membrane of the vesicle. And when the vesicle fuses with the Golgi, it becomes a part of the membrane of the Golgi. And when the Golgi is done processing it and glycosylating it, and it packages it up into a vesicle, it becomes a part of the membrane of the vesicle. And then that vesicle travels up to the surface. And when it fuses, the membrane of the vesicle becomes a part of the membrane of the cell. And so that membrane protein is already embedded. So I want to take a second and ask you this question. Does that make sense? Yes, no, or kinda?
I just have a question.
Go ahead.
This number 2 in that CD4 does not needing to go inside the lumen, then it just hang out in the membrane
Yes.
of the ER.
And one way to think, because this is not a molecular class and you don't necessarily have all this background, but when you put a protein in the membrane, it's just the middle part of the protein that's actually inserted in the membrane. Usually, not always, but usually there's a little dangly bit that's dangling out one side and another dangly bit that's dangling out the other side, if that makes sense. It's sort of like, imagine like a window screen and you stuck a pair of scissors through it, right? So the middle part of the scissors is actually in the screen, but there's like stuff dangling out each side. That isn't always the case, but that's a good way to imagine it. Does that help?
Yeah, I just don't get how some protein need to be inside and some just dangly.
Well, don't worry about that distinction too much, but just know that if you need it to be in the membrane, like whether it's dangling or not, just has to do with what it's going to do there once it's in the membrane. As it happens for CD4, you do need a part that's sticking out because CD4 is actually going to touch stuff, right? And you do need often something that's dangling inside because once it touches something, it's going to do something inside the cell. But there's other things that don't have dangly bits, but you can just assume they all do, right? That they're in the membrane for a reason, right? What is a membrane protein doing? Usually it's a receptor. So it's in the membrane, so it will stay on the surface of the cell, but it's sticking out trying to grab stuff. Does that make sense?
Right, because just because what they wanted to do, then that's where they position themselves.
Yeah, because whatever that membrane protein is doing, and in the case of CD4, for T helpers, what CD4 does is it helps the T helper to grab onto the MHC2, which is on the surface of immune cells. But there's lots of reasons for things to dangle outside. Okay, next. And I don't know if you notice a theme here, guys, and I don't know if you understand that it is deliberate, but I will call it out. I am talking about cells that are human cells that we will eventually encounter in the immunology section. So why not understand them a little bit better in this context? So the next cell we're going to talk about is a phagocyte. And phagocytic cells, of course, what do? They phagocytose things. It's a form of endocytosis. They go around gobbling up stuff. Sometimes this stuff is as big as a whole bacterium. Sometimes it's a virus particle. Sometimes it's a little toxin protein. But they're going around grabbing stuff and gobbling it up to destroy it so that it can protect you from to protect your body from whatever it is. Okay. So the phagocytes do a lot of phagocytosing, endocytosing, but they then have to digest what they have found. So they clearly need a lot of lysosomes. And lysosomes are made by the endomembrane system. What happens is, I'll sort of describe it to you in words. I'm going to send you away so you can do this activity to review this, but lysosomal enzymes end up in the interior of the lysosome, which is just a specialized vesicle with an acidic interior and lots of digestive enzymes inside. Those enzymes have to get there inside of the lysosome by going through the endomembrane system. The endomembrane system produces lysosomes. So the enzymes go through the ER, they go to the Golgi, and in the Golgi recognizes that those are not supposed to go to the surface of the cell. They're not supposed to be sent outside like antibodies or like insulin. They're not supposed to go to the membrane like membrane proteins. They're supposed to be in the lysosome. So it kind of gathers them all up together and makes a pouch and then just tells that pouch, don't go to the surface. You're just going to hang out in the cell and be a lysosome. So it's made just like a vesicle that's destined for the membrane, but it's a specialized one that's just supposed to be a lysosome. So with that in mind, see if you can figure out which of these in what order are required to make a lysosome with the endomembrane system. I'm going to send you to your breakouts now. So off you go. When you're done, come back, put your answer into the chat. That's why I'll know that people are starting to get wind down. Breakouts close in 10 seconds. As you're coming back in, if you put your answer into the chat, go ahead. All right. Welcome back, everyone. As you're coming back in, make sure to put your answer into the chat, at least as far as you got. If you didn't get it at all, you can always put a question mark. It's all good in the hood. Okay, so let's have a look at what the answer is and talk our way through it. Okay, so many of you got here, good job. So the first thing we have to understand is our goal is to get a bunch of enzymes into the interior of the lysosome. So we're going to start with one because we don't need to be in a membrane. We don't need to be in the plasma membrane. We don't need to be in any membrane. We need to be in the inside of the lysosome. So we can just make our protein in the inside of the ER, send it to the Golgi. And remember what the Golgi does is it then recognizes it's a lysosomal enzyme, doesn't need to get glycosylated because it's not leaving the cell, doesn't need to go to the plasma membrane. It just gets gathered up and sorted and packaged into a vesicle that becomes the lysosome. So it's that simple. Okay, now last but not least, what if we wanted to make an enzyme that's going to do the job of being in the Golgi as a glycosylation enzyme? So when you're in the Golgi, the things that are going to the surface, to the outside world or to the membrane, they need to be glycosylated. They need to be decorated with sugar so that stuff that's outside the cell has sugar on it. That enzyme or that process has to be accomplished by an enzyme. So we have to actually make that enzyme and it has to end up in the Golgi, and for it to end up there, it needs to be made by the endomembrane system. So I'm not going to send you away for this one. I just want you to think about what steps would be required to accomplish this. Put it into the chat when you think you know. Put a question mark if you have no clue, but let's see what we can come up with. So again, here's the answer. We make the protein in the interior of the ER and we send it to the Golgi, and then it shows up in the interior of the Golgi. Are these included on our exam? You have to understand this. You could be asked a question that will require you to understand this, but it won't be like this. It won't be like, here's possible numbered steps and what would be happening, but a basic understanding of these different, like how to accomplish these different goals is important. Any other questions, guys? Okay. Like I said, feel free to reapproach this at the end of class if you need to. We want to move on now to our final topic, or second to last topic, which is the mitochondrion and chloroplast structure. I just briefly want to review where stuff is so that when we get to metabolism, it is easier to understand. Okay, so these two organelles are the energy and carbohydrate generating organelles. And the mitochondrion has this particular structure where there's two membranes, an outer membrane and an inner membrane. And this creates two separate fluid-filled compartments. The very, very interior is the matrix. And then the space in between the inner and outer membrane is also filled with a fluid, and that space has a name too. It's the intermembrane space. Okay, so what do you want to know about your mitochondrion? You want to know that it has an inner membrane and outer membrane. You want to know where the matrix is, the most interior fluid. You want to know where that intermembrane space is in between these two membranes. What, if you know or want to guess, is the pH of the matrix? The matrix has a pH. What is it? When we get to metabolism, this will be somewhat helpful to know. And you can just ballpark it and say neutral, acidic, or alkaline. Yeah. The pH of the matrix is neutral-ish. It's slightly alkaline, 7 to 8. So it's around 8 if we round up, but it's close to neutral. And that's because there's a lot of biochemistry that has to happen. And you may recall that enzymes are pH sensitive. So most of them work best at neutral pH, though not the lysosomal ones, those like an acid one. But the pH of the matrix is close to neutral, and that's important for the function of things like the enzymes of the Krebs cycle. The electron transport chain is an important part of respiration. It's in the mitochondrion. Where is it located? The electron transport chain is a bunch of machines that are embedded in a membrane. Which membrane? There are two to choose from, the inner and the outer. Let's see if you can get it right. Correct. It is the inner mitochondrial membrane where you find the electron transport chain. Now, what happens is during respiration, protons are pumped from the matrix into the intermembrane space. And that creates a gradient. And that gradient is then permitted to be dissipated by allowing those protons to come back through ATP synthase, which spins the turbine and makes ATP. So protons are actively transported from the matrix to the intermembrane space, and then they diffuse back through ATP synthase into the matrix. Here's our chloroplast. The chloroplast is a slightly more complex organelle. It has 3 membranes, not 2. There's the outer and inner membrane, and then there's this network of membranes on the inside, which are the thylakoid discs. And this creates several fluid-filled compartments. We don't actually have any utility for the intermembrane space, so don't worry about that for chloroplasts. But there's the interior or the lumen of the thylakoid, that would be the inside fluid that you can't see. And then there's the fluid that bathes the thylakoids called the stroma, right? So we want to know where these two inner and outer membranes are, and then that the thylakoid membrane is inside of that. We want to know that the fluid that's inside of the outer membrane, but exterior to the thylakoid, is the stroma. What is the pH of the stroma? Noting that it has a very similar location and function to the matrix of the mitochondrion. Yeah. It's neutral-ish, right? For the same reason. There are enzymes that participate in biochemistry, notably the Calvin cycle, that must function, and their pH needs to be close to neutral. The photosynthetic process requires electron transport chains and photosystems. These are embedded in the membrane. And which membrane? You've got several to choose from now. Is it in the inner, outer, or thylakoid membrane where you'll find the electron transport chains and photosystems? Yeah, in the thylakoid membrane. Now, similar to respiration, there is ATP generation that involves a proton gradient. The protons are pumped, actively transported, in this case, from the matrix into the interior of thylakoid, where they accumulate, and that high concentration of protons is then dissipated by allowing them to run back through ATP synthase to generate ATP. Okay, so you want to kind of know where all this stuff is. And just a reminder, can prokaryotes do cellular respiration or photosynthesis? We've just described how those processes kind of work in the mitochondrion and in the chloroplasts, which are only found in eukaryotic cells. Can prokaryotes do cellular respiration or photosynthesis since they do not have either of these organelles? Yes or no? Great. The answer is, of course, yes. And since there's no inner mitochondrial membrane for the electron transport chain to be, since there's no thylakoid membrane for the photosystems and the electron transport chains to be, where do prokaryotes put all this machinery? That's right, plasma membrane. Okay. So this brings us to our last topic, which is the endosymbiotic theory. And this is a theory that explains how mitochondria and chloroplast are thought to have evolved and how they sort of contributed to eukaryotic cellular evolution. Let me ask you this question first. Who actually proposed this theory? This is part of your history section that you do need to know for the exam. Correct. It was Lynn Margulis. And then in a nutshell, what does this theory propose? Raise your hand if you can explain what it says. Gabby, go ahead.
I believe that it is that the mitochondria, sorry, my dog's barking. The mitochondria, or no, yes, and chloroplasts are formed from basically evolving bacteria into the cells.
Yes, exactly. So the origin of these organelles is thought to have originally been a symbiotic relationship between prokaryotic cells and an early eukaryotic cell. So the idea is that originally there were free living bacteria that could do respiration and free living bacteria that could do photosynthesis. Those bacteria still exist today, so we know that they existed. But those free living bacteria were engulfed by, endocytosed by an early eukaryotic cell, and they developed a symbiotic relationship where the respiration ability of the bacterium allowed the bigger cell that had engulfed it to stop doing that and just delegate that to the mitochondrion or the evolving mitochondrion, or delegate photosynthesis to the bacterium, which became a chloroplast. Over time, over evolutionary time, they had such a tight relationship, they stopped being able to live without each other. So there's several lines of evidence that support this. Write into the chat any of the lines of evidence that support this theory. What was it that caused Lynn to come up with this idea? Yes, the organelle's structure is that it has two membranes, just as if it had been engulfed by a cell. When you engulf something, you wrap it in a membrane as it comes in. So that explains the two membranes. Then the size, right? Their size of chloroplasts and mitochondria is similar to bacteria. The fact that these two organelles have their own DNA, and that DNA is circular, like a bacterial chromosome, not similar to the eukaryotic chromosomes. And they have their own ribosomes, which are 70S bacterial ribosomes, not the 80S eukaryotic ones. So it's their structure with that extra membrane, it's their circular DNA, their 70S ribosomes, and they have a couple of other features. Their size, their replication, when they replicate, it looks like binary fission. Okay, so here's a little graphic of that theory. Okay, so this is not how your unit test exactly is going to work. It's going to happen on a you're not going to go to Zoom. I'm going to sort of give you a heads up about the test now, though. You're going to come to your lab, and in lab you will do some lab. And round about the halfway point, which is 7 o'clock, we're going to migrate over to a computer lab in Chuckson Hall, and you will go into Canvas and open up the test in Canvas. And the test will be proctored. I'll be in the room. You can ask me questions. And you will be taking the test on the computer, but proctored. Now, a couple of things. The rules of this test are the same as if you were taking this at home. My expectations are the same. The test is open book, open note, hard copy notes. You are not allowed to Google anything. So you cannot have any devices with you that are connected to the internet, except for the actual computer you're going to be working on, which has a keyboard tracker. And so it will alert me if anybody tries to click out of the testing window. Okay, so while you're on that computer and you are connected to Canvas, you have to stay in Canvas. You cannot leave. So if you have hard copy notes, you can bring those to the test. Okay. Yes, you can bring your textbook and your notebook. You can bring a calculator, that is allowed. Okay. If you want to use your phone as a calculator, then you have to show me that it is in airplane mode and is not connected to the internet. All right, those are the rules of the game. And the rules are similar at home if you had to take this test at home, which in test 3 may well be the case. I have to check on my logistics there, but I'm most likely going to do it in Zoom. Could calculators be provided, Haley? That's a good question. The answer is no, but if you email me, I will investigate. I do not have any way to do that, but possibly I can find out and I may be able to work on that. So for right now, assume that you cannot, and that you'll have to bring your own, but email it to me and I will look into that. You can have as many sheets of paper as you want, Nikki. You can bring the whole textbook, you can print off 100 pages of notes, I don't care. It is not tomorrow. Oh my gosh. Thanks for asking that, Tao. It is not tomorrow. It is next Wednesday. Next Wednesday. On Monday, what we're going to do is we're going to actually have a Unit 1 review. What if a question is not on the study guide? Do we get points for that? Jenna, you can say more about that? I'm not sure what you mean. What if a question is not on the study guide? Do you mean if a question on the test is not on the study guide? Is that what you mean? I see. While I don't promise the study guide is 100% comprehensive, it's pretty much comprehensive of what I would expect to see on the test. My expectation is that you attended the Zooms and understood what we talked about, and that you watched the video lectures and understood what's in them. And the study guide is a pretty good representation of what is most important to focus on. So I would lean into that. If there's some particular part of the curriculum, like things I don't ask you to memorize ever what causative agents cause particular diseases, or what the symptomology of particular diseases is, or all of the lab procedures except for the ones we explicitly covered in class. However, problem solving things that we've done where there's math, you can guarantee that will be on the test. And any sort of general facts about microbes you need to know in the history that we covered. Does that help? It should be on the study guide, but might be a stray question. It would be something that was covered in video lectures in Zoom and in the lab, and should still be well understood. Okay. All right, I'm going to pause the recording then, or stop the recording.